Hi everyone, welcome to our webinar. My name is Rebecca Northeast and I am a product manager here at Proteintech. I look after the ELISA kits as part of my product portfolio. And today we are going to present to you our ELISA workshop, specifically focusing on best practice techniques in data analysis. I'd just like to say at the start that if you have any questions, I will get round to answering at the end if I can. Please write these in the comments section or in the Q&A box. We have Callie online as well, who is our technical specialist. She works with us for tech support and she will be helping me at the end with the questions. So she'll also be monitoring the inbox as well. So if you have any questions that you want answering now, hopefully Callie will be able to write them down. So let's get started. So firstly, I'll do a really quick company introduction. So in case you aren't aware of who we are, Proteintech is a leading life science reagent manufacturer of antibodies, immunoassays, which we sell through our Proteintech brand. And then through our Humankind brand, we sell our cytokines and growth factors. And these are recombinantly expressed in a HEC293 expression system. So these are human cell lines. So that means that they have native folding, post-translational modifications, and glycosylations. We have these in research use only and in GMP grade as well. So do be aware of that. We also have, through our Chromatech brand, our nanobody-based reagents. So nanobodies are really excellent for things like immunoprecipitation of tagged proteins, as well as for super resolution microscopy due to their much smaller size. As a company, we like to celebrate our success by celebrating our customers' success. Specifically, in that we've been cited in over 200,000 times worldwide, which was a really exciting milestone for us that we reached earlier this year. And last year, we were also awarded the Researcher's Choice Award by Citab. This wasn't anything that we bought or paid for from Citab. It was actually something that people like yourselves nominated us for. So we're really excited to have won that. So what am I going to talk to you about today? So briefly, we'll have a quick introduction to ELISAs, looking at ELISA best practice, ELISA data analysis, the difference between making your own versus a commercial pre-made kit, how we validate our ELISA kits, and then some basic troubleshooting if we have time at the end. So just a quick introduction to an ELISA. So ELISAs are essentially the gold standard for quantitative analysis of crude biological samples. These are a really quick and simple protocol and is very easy to automate. However, it cannot be used for spatial information. So if you wanted to look at the presence of a protein in, say, a tissue, where it was presented, where it might move in disease, then you'd want to look at something like immunohistochemistry. So ELISA cannot be used for that. It was originally first described or created in 1971 and now is really widely used in research and clinical settings. ELISA formats are in things like your LFT for your COVID. They're used to detect HIV in patients, as well as across loads of different research labs. So two of the most common immunoassay techniques for detecting a protein are the ELISA and the Western blot. But how do these differ? So they both allow really highly sensitive detection of an analyte or your protein. ELISAs can be quantitative as well as semi-quantitative, whereas Western blots are more qualitative. One of the best things about ELISAs is that they're really quick, simple, and high throughput, and they allow for you to detect a protein or analyte in a crude sample preparation, so you don't have to create a protein lysate, for example, in order to then run it like you would in a Western blot. But one of the downsides as well is that you only get concentration and presence of the target, whereas Western blot allows you to detect post-translational modifications, any mutation in your protein during disease, and also its relative expression. So there are 4 types of ELISAs that are typically used. So the first and the easiest is typically your direct ELISA. This is where you coat your antigen to the plate, you add an antibody with a conjugate, and then you add the reactant, and then you see how much analyte is present after it's been coated to the plate. For an indirect, it's pretty much the same, but you're using a secondary antibody, so you get a little bit of amplification there, allowing it to be a bit more sensitive than a direct ELISA. And then for your sandwich ELISA, these are really specific and sensitive, and that's because you use pairs of antibodies. And essentially what this does is you have a coating antibody and a capture antibody and a detection antibody, and that allows for higher specificity. And I'll talk a bit more about that in a second. And then lastly, we have our competitive ELISAs, and these are really good for quantitative measurement of low abundant analyte. And essentially what happens if you have a inhibitor antigen, and then what happens is that the higher the presence of your actual antigen, you then get an inverse proportion of signal. But today, we're going to focus on the sandwich ELISAs because this is predominantly what most people use, and they are the most specific and sensitive, so are kind of considered the gold standard of ELISAs in the industry. So what is the standard ELISA process? So this first starts with by coating your plate with the capture antibody and then washing and then adding a blocking reagent. And this is done if you have a commercial kit, this is really done for you. And then you need to add your samples, your controls, and your standards. Then adding the primary antibody, and that primary antibody might then have the detection enzyme as well, or sometimes you might need a secondary antibody, which then has a detection enzyme. You would then add the substrate, and then you would perform the reaction that allows signal detection. And I want to say that using antibody pairs is really important in this standard ELISA process. It actually allows to increase the sensitivity around 6 times than a traditional sort of indirect ELISA. And also it enables more specificity because you have 2 antibodies binding to the protein at different epitopes. That means that you have to have you are more likely to detect your analyte of interest rather than have any off-target binding. So now we're going to move into ELISA best practice. And so ELISAs are actually quite simple to run. However, there is quite a lot of room. There can sometimes be some room for error, and that can be done through things like not obeying the correct temperature and time, or perhaps having inconsistent pipetting technique. And one thing to remember is that ELISAs can be really reproducible, and if you just think about them as a chemical reaction. So we all know that reactions are influenced by things like time and temperature, as well as binding kinetics and the amount of substrate availability. So this is why time and temperature incubation is so important. Your pipette technique is so important, and the sort of availability of the reagents and samples, because if these aspects change, then the readout would change and you're just going to have a slightly different result. It's not like IHC, immunohistochemistry, where you can see where something's gone wrong. You're just getting a simple biological readout here. So you want to make sure that everything you're doing is correct. So one thing that we can do when running an ELISA is always to plan ahead. First, you want to ensure that you have enough of each reagent available to complete your planned experiment. And if you have multiple kits or multiple lots of buffer, for example, do not mix them. It's really important that you want to keep everything as standardized as possible. So if you do run out of something, it's okay if you want to mix between those lots or between the plates of the same lot, but whatever you do, do not mix a lot, especially of things like buffers and antibodies. Next thing you want to do is to make sure you're calculating your reagent and sample volume needed, and then decide on if you're running on duplicate or triplicate. It's pretty much gold practice to run in at least duplicate so that you can assess for pipetting error, and then therefore avoid that sample if needed, if you think there's a pipette problem. One thing as well is you want to complete your dilution calculations before sampling. And if you are worried that your sample of interest is going to be outside of the limits of your antibody binding, then make sure you do a test plate to check the dilution of your samples if needed before running a whole experiment and potentially wasting everything because you're outside of the read. So next step is looking at how we plate our samples, reagents, and standards. And one thing that I want to stress here is how important it is to make sure that your reagents are warmed to room temperature before starting. You want to get them out on the counter for at least 30 minutes before you start, allow them to equilibrate to room temperature. And this is actually because many of the reagents contain temperature-dependent components that might come out of solution as still cold. So as you can imagine, that would really affect the ELISA reaction. When you're using your reagents at room temperature, this means that you will have consistent binding kinetics and also consistent color development from one assay to another. Therefore, it's really important that you always run things in the same way, always ensure that you've left sort of adequate time for reagents to come to room temperature so that you then can compare your samples between one and the other. As I said before, run everything in duplicate or triplicate for the same plate. Use fresh standards every time. At Proteintech, we provide at least 2 bottles of standards so that you're making sure that you can, if you're running the plate twice, you're using fresh standards when you're running the standard control. Include blanks. This might be sample buffer or any other buffer that you're using. Sometimes it's also necessary to include positive controls. So say you're running something in a sample or you've done an experiment type that you're not quite sure what's going to happen, if it's going to reduce your sample too much or increase it too much. By running a positive control or something that you know is supposed to have a certain amount of that analyte present can really, really help you interpret your results later on. And it will also help you assess whether or not it's your sample, it's your pipetting, it's your experimental technique, something else that's gone wrong there, rather than just necessarily the real biological result. One other thing to note as well is really always produce reliable standard curves and do not expand the suggested range. And this is because the upper and lower here asymptotes of the curve are limited by the unique characteristics of that antibody pair in your plate. And trying to change them, so for example, trying to increase the range that you might be able to have here will not change the kinetics of that antibody and will just end up with inaccurate results. So make sure that you perform the standard range as suggested by the manufacturer. We've spoken a lot about pipetting, but we really want to focus on pipetting because actually poor pipetting can cause huge result variability. So there are many different videos out there if you're unsure of how to pipette for an ELISA, and I highly recommend you watching them because it can really help in generating accurate, reproducible results between plates and time after time. So one point of note is to pipette at an angle and to not touch the bottom of the well. If you're touching the bottom of the well, you might be introducing bubbles, or you might be scratching off some of the coating that's on that ELISA plate already. Use multi-channel pipettes where possible because this is again going to try and eliminate user error or pipette error if you're having to do one at a time. And use new tips for different standards and different samples. This is kind of obvious. You don't want to be introducing contamination. You also want to try and avoid bubbles where possible. And one way to do this is to avoid blowout. So don't go all the way down when you're using your pipette. Other things is that you can try reverse pipetting for viscous samples. If you're unsure what reverse pipetting is, again, there'll be lots of videos out there, but it can be a really useful trick for not introducing bubbles into your standards, into your samples, and also because bubbles can be really bad later when you're trying to read out because it can affect the wavelength of the optical density when you're reading out your plate later on. Next is washing. So washing is really important in our ELISA. I've said really important a lot, but again, I think pipetting and washing is a really important part of our ELISA workflow. And this is because poor washing can be a common cause of high background, reduced sensibility and variability. If you are lucky enough to be able to have an automated plate washer in your lab, say you're a lab that goes through a lot of ELISAs, then definitely I would use this. Or if you are a lab that has a lot of ELISAs and you don't have an automated plate washer, see if you can invest in one because it will A, save you time, and B, it will also really increase the reliability and decrease the background of your ELISA kits. You can either do this manually, or if you have a plate washer, always add a 30-second rest period once you've added the wash buffer, so a sort of mini 30-second incubation before then replacing the wash buffer and doing another wash round. If you are doing it yourself, make sure you're tapping really firmly after washing so you're getting rid of all the wash buffer and trying to prevent any bubbles. And if you are using wash bottles and they have these quite small tips, you can cut the tips off just a little bit further up. And what that will do is help to prevent any bubbles entering. And again, we want to really minimize bubbles because that can affect the readout later on. As I said at the start, we always want to follow the suggested protocol for incubation times. So this is things like temperature time and whether or not you put it on a shaker. These are highly important because you want to ensure that your reaction is reaching equilibrium and the binding kinetics are consistent for reliable results. Always make sure you're using the provided diluents and buffers because these have been predetermined for that sample type, for that kit, for that antibody pair. You don't want to be moving around buffers if you have kits from different manufacturers, for example. Always use the buffer that's been suggested by that manufacturer instead of moving them around, swapping lots. I can't stress how much antibody binding kinetics can change if you're then choosing different buffers or moving them around. When our team is developing antibody ELISA kits, they're always really specific in generating standard curves and everything in that specific buffer because they know how much it can change. One other point as well is not to stack plates. Stacking plates when you're doing, say, a cold or a hot temperature can cause something called edge effects, and that's where the reading around the edge of the ELISA plate can be different, say slightly too high or slightly too low than in the middle, than the results in the middle. And that's because if you're stacking, then you're not allowing the temperature to either fully change throughout the middle. So if you are doing a warm or a cold incubation, make sure your plates are separate on the rocker or whatever, so that you can make sure that you're getting that even distribution of temperature and getting that even equilibrium of your reaction to ensure reliable and accurate results. So now moving on to data analysis. So first off, with ELISA data analysis, we want to create a standard curve. But why do we use a standard curve in ELISA kits and ELISA data analysis? This is because we use a set of standards at a known concentration. We can generate a line of best fit, and then we can compare the readout from our samples to that curve of best fit, and then we can pretty much estimate or extrapolate the value of real concentration for that. The standard concentration should range from almost undetectable to that maximum signal detectable by that pair of antibodies. So what would happen is that the curve would then flatten out. You're not going to have an exponential curve. And what this does is this therefore allows really great flexibility of the assay to try and detect sample analyte at a range of concentrations. We will then use a statistical model to best fit a predictive standard curve to then help us interpolate our sample values. So in terms of samples and standards, as I said before, run these in duplicate or triplicate, but you also want to understand your pipetting precision by calculating the average and standard deviation between your runs, so between the duplicate or triplicate, and then you can calculate the coefficient of variance. I put the little equation here if you're interested in that. And you want to make sure that it's less than 10 to 15%. And this is because then you're aware that anything greater than, say, around 20%, this could be caused by pipetting errors, contaminated plates or reagents, or perhaps even uneven temperature across a plate. Like I said before, if you've been stacking your plate, that could cause a much higher CV, coefficient of variation. Always run a standard curve for every plate. I've had people ask me this in the past, and it's not something that's done by everyone, but we really recommend it here at Proteintech that by running a standard curve for each plate, you are accounting for sort of minor changes in temperature or pipetting or things happening that day rather than relying on a standard curve that was generated separately. As I mentioned before, try using a positive control with known concentration, and always include blank sample wells because this will help determine background absorbance, which we will then subtract from all our readings later on. One point of note here as well is that you have to try and dilute your samples so that they fall within the range of the standard curve. Like I said before, the antibody kinetics have been designed to specifically bind within a linear range within that curve that's been suggested. So if you have samples that are perhaps falling under or over, you can't specifically say that the reading will be accurate to the actual biological concentration. So ensure that you're always trying to work within that range of the standard curve as provided in the kit. In terms of selecting the best curve fit for our standards, one thing that you can do to look at this is looking at the R-squared value. So the R-squared is a goodness of fit measure for sort of linear regression models. And so this most standard and simplest linear regression is simply this one here, where it's essentially like a straight line. But as we know in biology, a lot of the time, real biological data doesn't necessarily fit a linear curve fit. So other ways that we can look at things are semi-logs. This is where you are logging the concentration. And this has a bit of a better fit. However, what it does is it provides a sigmoidal curve. And then you can also do log-to-log fit. But again, this is okay. It's better than the standard linear regression, but it does compress some of the data points again. So it's not as easy to try and read off the curve. At Proteintech, the curve fit that we suggest is best is something called the 4-parameter logistic curve fit, or 4PL for short. And this is really widely known as best for fitting data from a biological system. What this does is it estimates 4 parameters that kind of help fit the data around a certain point. And this model will essentially have an S shape if you expand all the points here, but it essentially creates an almost linear line for your standard curve here. And you can see with the others that R was less than, not as good at being nearly closer to 0.99. But here you can see that R equals 0.99. So you can see that this is a much better model for fitting our data here. And one point of note is that if you're not sure which curve fit to go for, then it's really recommended that you back calculate your standards. So what we do here is you then sort of fit the standard curves to your fit the standards to your plot the standard curve, and then you would then back calculate. So you would treat the standard values as unknown, and you would interpolate their OD values to the standard curve that you just generated. And you want to make sure that they're reading really close to the expected values from the curve that you've just generated with them. So that should be around sort of 5 to 10%. If you're not getting that, then do try a different method. There's also something called the 5-parameter logistic, or if you think that a log-log or semi-log plot would be better, that's one way to check here. So I just wanted to let you know that our scientists at Proteintech have come up with our own free curve fitting software. So this is available online. And it's something that there are many other curve fitting tools online as well. And I really highly recommend them. In terms of when you're trying to plot yourself. It's actually really complicated to try and plot a standard curve, especially a 4PL or a 5PL, not using a specialist tool. I know GraphPad Prism does this, and I think it's what I used in the past, way back when I was back in the lab, but I know that GraphPad you have to pay for it. So along with Proteintech and also there are other websites as well, you can use our curve fitting software in order to help you do this. So this is an example of a screenshot from our curve fitting software. So you can see, you can just simply paste in your OD data, and then it's quite flexible. So you can decide how many standards you have, where the controls are, what your replications were, and we've got it to fit either 7, 11, or custom standards. Typically, most standards require 7. So you've got this here, and then you would simply set up your initial concentration, say which way it's going, if you're dividing or timesing, and then it will come up here. Once you click submit, you'll be able to check that these are the correct standards that you used. Also, along with our curve fitting tool, because we know that the 4-parameter logistic, the 4PL, is the best curve fit, the default setting is 4PL, and then also logging the X data and the Y data, so the so the OD and the concentration. And also we've done it to automatically deduct the background. These can all be played with. You can change your regression fitting model to say something like a linear regression or a semi-log fit. And you can also have the flexibility of naming it yourself. And then once you've generated the tool, you can then use your standard curve to extrapolate your sample concentration from the OD value. So you've got your readout, you've got your OD values. You then need to use your standard curve in this sort of traditional way to then read out your concentration. In our tool as well, we do enable this for you. So you can go on to the next page and then assess your concentrations value that way, or you can do it yourself by simply hovering the mouse over the curve and then getting the readout that way. So onto just simple data analysis. Once we've got the standard curve, what you want to do is subtract your background absorbance from all data points. So the background absorbance that we mentioned before, these are your blank well values. So that's probably using just standard or sample buffer and nothing else in it. You want to average those blank wells, and then that becomes your background absorbance. And then you want to minus that, subtract that from all your data points, including your standards. Make sure that you use your standard curve to extrapolate the sample concentration from the OD values. That's what we showed you in the slide previously. And one point that's really important as well is to remember your dilution factors. So say you're diluting your human serum sample by a factor of 4. Once you've got your OD value, make sure you multiply that by a factor of 4 again, so you're multiplying by 4, so you're getting the actual concentration. Otherwise, the readout that you'll get will be slightly lower and not accurate. And as we said before, calculate the averages of your replicate. So that's if you say been doing it in duplicate or triplicate, calculate them in standard for the standard deviation and the coefficient of variance to ensure accurate pipetting as well. So now we're going to talk a bit about the differences between making your own kit and commercial kit. So now being an ELISA product manager for 2-3 years, I'm now so aware of how complicated it is to actually develop an ELISA either commercially or not commercially. And this is because there are so many different areas of consideration. So one of them is, there's 4 key ones here. So you first want to think about the immobilization of the antibody. Is the antibody binding in an orientation that means it can then it can recognize the analyte? Is the material that you're using, so the type of the plate, is that inactivating the antibody? Is it causing high background? And does it have high enough binding capacity for your antibody of choice as well to then recognize the epitope? And then in terms of antibody design, you want to make sure you've you've got an antibody pairs that are looking at non-overlapping epitopes. And trust me when I say finding antibody pairs is really quite hard. You've got so many different clones and epitopes you can need to think about. You want to ensure that by binding one antibody, you're not hindering the site of the other antibody. So like I said, finding antibody pairs that work in an ELISA is pretty much all the work that goes into antibody commercial development, ELISA commercial development, sorry. Then you've got to think about your conjugation or your labeling strategy. Are you going to be using a directly conjugated detection antibody, or are you going to be using a secondary antibody that has the detection reagent itself instead? And then once you've got these pairs, you want to think about antibody and sample dilution. So you can do what's called a checkerboard titration. So this is where you assess two variables at once. In this case, it would be looking at antibody concentration and sample concentration, and then you can essentially assess the best ratio of sample to antibody concentration by running it like that. You want to look at sample spike and recovery, and we'll talk about that in depth in a couple of slides' time. But essentially, everything that you're doing when trying to develop an ELISA is to get the optimal signal to noise ratios. You want to ensure that you're getting enough signal from the background noise of your kit. And the last point is our block and wash buffers. You can get a lot, you can purchase these commercially, which I probably recommend if you're developing your own ELISA, or if you're making them yourself, you want to make sure that you're quality controlling them well. If you've got the salts and detergent at the optimal pH for that antibody binding, and is there minimal enzyme activity in there as well. So as we know, ELISAs are essentially a way of utilizing antibody pairs to sensitively detect and analyze. So in order to do that, we want to look at matched antibody pairs. So this is whereby you have two antibodies that bind to a protein at a different epitope. And you want to ensure that these antibodies are well validated. So ensure you're inspecting the original validation data. And how reproducible are they? How long-term is this project going to be? If you're looking at something that's going to be a really long-term project, then you kind of want to focus on the recombinant and the monoclonal antibodies over the polyclonals. If you are developing your own ELISA and you're really struggling to find matched antibody pairs, companies like Proteintech, who make every antibody themselves, we have freezers full of clones. So if you are really trying to do this, then do reach out to us. We might let you to try some different clones that we have available in order to develop these ELISA kits that aren't necessarily commercially available, but we would be able to work with you in that sense. If you are using polyclonal, then try and ask for lot reservation. We do that for some customers if they're making kits, or if they have a specific lot in mind that they find works best for them. We are happy to reserve a certain number of vials of lots for them for, say, 1 to 2 years. And then, as we said before, using that checkerboard titration, you can determine the best concentration of antibody using a serial dilution. So a lot of customers have said in the past, like that they much prefer making their own because of cost. And we totally understand that at Proteintech. We understand that grants are precious. However, as we just showed, it can be incredibly complex to try and make your own kit. So overall, purchasing a pre-made kit will hopefully save you money in the end. And there are many, many benefits as well. So you've got matched validated antibody pairs and standards included. Every step will be quality controlled. So every company that provides antibody ELISA kits, they're ensuring that there's high levels of consistency, accuracy, and reliability, checking between lots, checking between plates. You're removing a lot of user variation by purchasing a pre-coated plate already, and also by purchasing those where you know the antibody dilution and the buffer composition that's all been checked for you previously. Having recommended sample type dilutions, this will help conserve samples. It will help save you a lot of time as well. And also by a lot of ELISA kits like Proteintech's come in a strip plate format. So this enables you to test samples before running the whole plate, so you're not wasting anything. So in terms of the Proteintech ELISA kits, we have over 350 kits and targets available. We're ensuring that these have extensive quality control and time in development. I work with our team all the time for this, so I can really attest for how much work they put into ensuring that all our kits are valid in relevant sample types, that they're providing accurate and reliable data for you as well. We validate these all in-house, and we're ensuring that we're doing it in natural samples. We're not just looking at spike protein in buffer, for example. We're doing this in natural samples using natural matrix, so things like serum, saliva, cell lysate, things like that. As mentioned, they're in a strip plate format, like you can see here. So this means you can save some strips and run a plate 2 or 3 times. That means that you don't have to, that means that the slightly more costly plate you can use multiple times. So it's actually saving you time and money in the long run. Tech support is directly with the R&D scientists. So if you have an issue with the kit or you aren't sure of how your results should be, if you send us an email, the people you'll be talking to are some people like myself and Callie, and then we liaise directly with the scientists as well to get the best answer for you to try and get you the best results. We're ensuring that the sample dilutions are predetermined to try and help you if we validated it in that sample. And also, we are 50% cheaper than leading competitors. And that's because we really believe that accurate and good results shouldn't cost your grant loads of money like we know it does from the competitors. If you are interested in a Proteintech kit, then you can try one for $99 using this promo code, or at the end, we'll send you a form in which you can submit and we'll be able to send you a quote to then get the ELISA. As well as our standard Proteintech kits, we also sell our Authenticine ELISA kits, and these are made using our humankind proteins. So our humankind proteins are growth factors and recombinant growth factors and cytokines made from HEC293 cells. So what this means is that the antibody pairs that are used to as immunogens to generate, these humankind proteins that are used as immunogens to generate the pairs for Authenticine, they can actually recognize proteins that have native glycosylations and PTMs. So that makes them more sensitive because they're recognizing all those different glycosylation environments. And they're actually can be between 10 to 1,000 times more sensitive than leading competitors. And that means that you can look at low volume or precious samples, so you can use a lot less volume, or you can look at a low abundant analyte by using an ELISA kit where you previously thought you couldn't. And again, these are under the promo. So if you are interested in trialing our human Authenticine ELISA kits that are extra sensitive because they really recognize human proteins in their natural environment, then yeah, do think about trialing one of our Authenticine kits. So now I'm just going to talk about ELISA kit validation. So in terms of how we validate our commercial ELISA kits, first we want to look at our antibody selection. So we said this before, validating pairs is really complex and long. So ensuring that we're identifying clones that are compatible and not interacting when they bind the analyte. They're ensuring that we've got the best clones for the situation here. And we also want to ensure that they're compatible with a detection antibody and conjugating that detection antibody to HRP or biotin or any other kind of detection enzyme. We're then assessing the coefficient of variance between inter and intra variability for every batch. I believe we test at least 24 plates in this way. We then do spike recovery, and I'll talk about this in the next slide because I think it's really interesting and why we should do spike recovery for our ELISA kit validation. Same for linearity and range. This allows us to test our assay in various different sample dilutions and also test assay precision, but I'll talk about that in a minute too. And then sensitivity. So sensitivity is the theoretical lowest amount of analyte that you can test using that kit. And the way that you do that is you look at your average blank value and you times the 2 times standard deviation of that blank value to reach the theoretical sensitivity of that kit. Other things that we and other companies do is that we do natural sample and parallelism with our commercial kit validation. And what this does is that we're looking at increasing concentration of natural samples and ensuring that these are replicating the standard curve, showing that our kits work not only for recombinant protein, but also for detecting samples detecting the analyte in samples as a customer would in its sort of natural sample matrix. So I just briefly mentioned spike and recovery, but why do we want to do this for an ELISA kit? So spike and recovery is done during an ELISA kit validation because it helps to assess the effect of the sample matrix on antibody binding to an analyte. And what we mean by the sample matrix is everything else that's present in that sample other than the analyte itself. So there's all sorts of other things like complement factors, other antibodies, other things that are going around, especially in things like matrix or more complex samples. And these sample matrices, they can have a really big impact on how that antibody and how the antibody kinetics are working. So what you want to do is to assess this sample matrix. You can then spike a known amount of recombinant protein into the sample matrix, and then you want to compare that to the standard curve. So this is the recovery part, and it must be within 80 to 120% of the expected values. If this fails, if say you've got a much lower reading than expected, then you want to try higher dilution, because then you're going to be minimizing the effects of that sample matrix by diluting it out. So a simple example here is that if you have a serum sample that's spiked with a solution with a known concentration of 100 picogram per mil of recombinant, say IL-2, but the spike sample is only measuring at 80 picogram per mil when you're looking at it in the ELISA, then you know that there must be some factor in the serum that's maybe inhibiting the detection of the IL-2 by antibodies that are then used in that assay. So that's why we really recommend diluting to try and reduce any matrix effects. The next part of the validation is something called the linearity of dilution. And this is where you want to serially dilute your sample in the chosen sample diluent and then spike it with a high concentration of recombinant protein. So you can see that here we've serially diluted, I believe this is human plasma or human serum. And then we're looking at whether or not it's showing a linear dilution relative to the standard curve. So as we know, biology isn't linear. So you want to make sure that instead of just checking that you're getting a sort of linear readout of the absorbance, you want to backfit it to the standard curve, ensure that this linearity is relative to the standard curve. Acceptable linearity over a wide range of dilutions indicates that the assay can be used in a flexible manner, and it can be used to identify and analyze at a range of concentrations. Something that's quite interesting is that sometimes matrix interference will only occur if a factor is present at concentrations above a certain threshold. So that's why you want to ensure that you're looking at samples that have been serially diluted, because then you'll be able to assess whether or not at what point that matrix starts to interfere with the reading of that, with the binding kinetics of the antibodies. So that's why we perform our linearity of dilution. So next is the importance of sample type. So as we just showed, talking about spike and recovery and linearity of dilution, and doing this in various different sample matrices, if you're looking to purchase an ELISA kit and it's in an unvalidated sample type, we can't show as a manufacturer, we can't tell you, we can't guarantee its performance, because there are so many different things in different matrices that will affect antibody performance, antibody kinetics. So as I said, quality control for commercial kits is rigorously done for different sample types. So say you purchase an ELISA and it says it's valid for serum, cell lysate, and saliva, but you want to look in fecal matter, hence my funny little emoji here. One thing that you can do is you're welcome to still purchase the ELISA, but what you might have to do is perform some other experiments yourself, because you want to assess that the matrix has not changed the assay performance, which would then lead to inaccurate results. So you'd want to perform spike and recovery and linearity of dilution. So if you have to do this yourself, I didn't have time really to go over the data analysis that you would need to do for that, but there are lots of really excellent protocols online if you do want to check the spike recovery and linearity of dilution of an ELISA kit, or let me know and I'll be able to send you the protocol. So nearly at the end, I'm aware that we've been going for 40 minutes, so I'm just going to really quickly go through some common troubleshooting and then maybe get round to a couple of questions. So first, you've got high signal. So I know this can be quite a big problem for ELISA kits. So like we said before, running an ELISA is simply just a chemical reaction. So if you do have high signal, it might be simply that you left your plate too long before reading. So once you've added the stop solution, you want to really try and read that ELISA within the next 5 minutes. If your sample readout is greater than the higher standard, then you've clearly exceeded the limits of the antibody kinetics. So you want to try diluting those samples in sample buffer to get them back within that ideal range of your samples. If you might have contaminated glassware. And one big problem as well is if people are trying to save money and using plate sealers, what might be happening is they're just introducing contamination. Next common thing for troubleshooting is just out of range. If your analyte is below detectable levels, then you can try more sensitive assay like our authenticine assays. Like we said before, they can be 10 to 1,000 times more sensitive than your traditional ELISA. Or there are other detection systems. So typically, it's not a chemiluminescent. So you could try a chemiluminescent detection system. You can get highly sensitive chemiluminescence as well that enables you to detect far lower than you could previously, say, using colorimetric. If it's out of range, then it might be that you've chosen the incorrect dilution. So do look at what your the manufacturer suggested. However, that is just a suggestion. So do try serial dilutions yourself before you then find the one that works the best. It could also be contaminated buffers or just, again, poor pipetting technique or other similar human error issues. The next one is high variation. And again, I think this is probably most common alongside high background. And this is typically due to poor pipetting or the poor pipette calibration itself. So try using a multi-channel pipette. Try using a automated plate washer, or change the way that you wash your plate, ensuring that you've got sufficient plate agitation, or you're leaving that 30-second rest that I recommended once you add your buffer before you then wash it off. Could be due to crosswell contamination or also bubbles in wells. If you do find bubbles, then there are ways that you can pop them before reading. You can use a little pipette and try and push them to the side. But again, try and avoid bubbles. We've spoken about that earlier in the presentation. There are other techniques you can do when pipetting to try and avoid bubbles. Other things with variation is, like I said before, is if you're stacking your plates during kind of warm or cold incubation, this can cause edge effects. So ensure that you're not stacking your plates and ensure that you bring up your reagents to room temperature before starting. I think we've just talked about high background, and now looking at matrix effects. So these are what we're talking about when we were looking at our commercial kit validation by doing spike and recovery and linearity of dilution. As we said before, the matrix, which is essentially everything else in your sample that isn't your protein, can have huge interactions with your antibody binding kinetics with your plate, for example. So pretty much the way that you can help get out of this problem, if you think that you have a matrix effect problem, is to dilute your sample in sample buffer, and then not forget to take that factor into account when reading it out. But like I said before, if you think matrix is a problem, if you're choosing a sample type that hasn't been validated by your ELISA manufacturer, then try running spike and recovery and linearity experiments to see how that's affected, and then try diluting to see if that makes it any better. So that was a relatively long webinar. I'm amazed that so many of you have stuck around. So the summary essentially is that ELISAs are pretty simple to perform, but really require consistency. Make sure you follow the best practice tips for accurate and reliable results. So that's things like ensuring that your reagents are warmed up to room temperature before starting. If you are performing data analysis and you haven't before, try our online free curve fitting software. This will hopefully really help you out rather than trying to figure it all out in Excel, for example. It's totally fine if you want to make your own kit, or if you're kind of doing a homebrew situation. But if you are just running the odd kit, commercial kits are extensively validated and can save you money and time in the long run. So do think about that in the future if you're trying a new, just say like one or two runs for a new sample or a new analyte that you're looking at. I can highly recommend commercial kits. Oh, and I also forgot to say that we made this poster. And I will send a link for you to request for it to be printed and sent to your lab if you wish. So you can remember everything that we said before about bringing your reagents to room temperature, about not stacking your plates. So you can hang this on the wall if you are a lab that does a lot of ELISAs. I think they look quite nice. So we'll definitely send you a link so that you can request that. And also, I just wanted to remind you that you are able to try a ELISA from us for $99. These are for customers in North America and Europe only, so apologies for that. But you can scan this form or you can use this promo code, and we'll also send you the form to everyone that attended in our follow-up email as well. And yeah, thank you so much for listening. I believe Callie will soon jump on, and we'll see if there are any questions that we can answer within the next sort of 5 minutes.
Hi, Rebecca. Thank you. That's a really excellent webinar. I learned a lot myself there. We've had lots of people online for the duration. We still have a lot of people online now, and we've had lots of excellent questions. Definitely too many to get through here, but I will pick and choose a few because we've had a really broad range of questions. So one of the questions, I guess, in terms of the sort of practical elements of the assay, someone has asked, is it safe to use a shaker during some of the incubations, even if it's not suggested in the protocol?
I would say if it's not suggested, then you probably don't need to use the shaker. But I don't think a shaker is probably one of the minimal things that will affect your response. If you are using a shaker, then maybe cut down your incubation time by, say, 10 to 20 minutes. But I'm not sure if it doesn't recommend a shaker, then hopefully you can find a non-shaky space in your lab to do the incubation. However, I do understand that sometimes, lab space is really really precious. So do try and always adhere to the manufacturer's recommendations.
Okay, great. So I had another question here, which was, if you know that the analyte is, there's going to be a lot of it in your sample, instead of dilution, could you shorten the sample incubation time or the secondary antibody incubation time
I would
as a way
definitely
to
not.
shorten the time of the experiment, which we all tried to do?
Unfortunately, I don't think that would work. I think antibody binding can happen within a matter of minutes. So I think you would essentially just end up with a really high readout that would go over and above that traditional linear range that the assay has been developed for. I would highly recommend just diluting your sample as much as you can so that it falls within that assay range. I know we're all trying to save time, and in the future, I believe that Proteintech is also developing things to try and save time, either through specialist labeling or through kind of like it changing some of our ELISA kit so that they are a faster readout. But for now, unfortunately, I would definitely stick to the recommended time and incubation. I'm afraid that's a bit of a boring response, but as we said, as like Callie and I know that we both come from the lab and we're really unsure how antibodies worked, and we were trying to, when we were in the lab ourselves, trying to get around certain things. But now we're kind of on the more technical commercial side. You understand how fast antibody binding kinetics are. So just by simply thinking that you can get away with it by reducing the incubation time, unfortunately, I think you would still get the majority of the antibody binding there and also the majority of the signal occurring so that you would go outside the standard range, standard curve.
Great, thank you. I have another question. Tricia has asked if have samples at both extreme ends of the standard curve limits, is it possible to use different dilutions in the same plate or to ensure that they fall within the standard curve range, or should I run two separate plates?
No, you can definitely dilute differently. That's totally fine. Just obviously make a note of which ones you diluted so that when you're coming back to calculate your experiment, to calculate your concentration later, make sure you're accounting for those different dilution factors.
Great. I have another question about ELISA washing. So which buffers would be recommended? And they've asked, would this be PBS or TBS, or would you recommend adding any detergents like Tween to the washes?
I can't remember off the top of my head, but I think yes, people do often add detergents like Tween for their ELISA washes, but I will have to check that. I think because I haven't got the protocol in front of me right now, sorry. I don't want to tell you the wrong thing necessarily.
No, and then I guess if you buy the kit, you will have the wash
Yeah,
buffer
yeah. I'm trying to
included
remember
in
what I
there, so
did
it'll be
way back
standard.
when, and I think we used Tween, but I don't want to tell you the wrong thing.
Okay, and then we had a few questions about the analysis and choice of method for creating the standard curve and why the 4PL is preferred over the 5PL. Is that across the board? Is the go-to to start with and then look at other options, or?
Yeah, that's a good question. At Proteintech, we always use 4PL, but I am aware that there are other ELISA providers that are now starting to recommend the 5PL. I think what you can do is if you have a software that allows you to do both, you can fit both and then do what I mentioned before, which is where you backfit your standard, the OD of your standard. So what you would do is fit both the 5PL and the 4PL, see what R value you get, what R square value you get, and then backfit the results from your standards from both of those, and then see how close you get. And the closer you get, so like the lowest percentage of difference is the one that you should choose. So I did speak about backfitting your ELISA standard curves to check which one, but that was more for people looking at whether or not sort of like a semi-log fit in a 4PL fit would work. But 4PL and 5PL are very similar. I think 4PL is based on the fact that it's a kind of symmetrical, whereas 5PL is based on the fact that it's asymmetrical. And some people would argue that the 5PL is better because biology is rarely asymmetrical. However, we found that the 4PL is slightly simpler and also in the majority of cases is perfectly fine for fitting ELISA standard curves.
Right, and I guess at that point you've got your numbers, so you can play around with the analysis and see what looks best.
I mean, you've got the numbers from the standards already, so there are different things that you can play around. But obviously, we're trying to be reproducible, reliable scientists here. So you don't want to run one plate with a 4PL because you think you get better results, and then the next plate with a 5PL if you're looking, if you're trying to compare between. Always make sure that you're using the same analysis between the comparisons.
Definitely. So we had a number of different questions about dilutions and matrix effect. A lot of them were quite specific. So I think that what we can do is have a proper look at the questions and we can get back to those more specific questions individually, as they're not probably going to be as useful across the board for different attendees at the webinar just now. So I think we could wrap up there in terms of the question.
Yeah, thanks so much for attending, everyone. At the end of this, there is a short survey which will just sort of help us tailor our marketing and tailor our products by understanding what our customers are doing. So thank you so much for attending, and I wish everyone a really good Wednesday. Bye.